Um, so again, thank you so much for inviting me, and indeed I too, which we were in Jerusalem. Thank you everyone from Israel for coming all the way here and for still being awake through these 2 days despite jet lag. So I'm going to talk about, continue on the theme that Gad actually brought up about whole genomes. So I'll talk about AI frameworks for interpretation of whole genomes, and I guess also continue on the Moses theme of really thinking about mechanisms and bringing that to disease insights. My group really thinks we're very much a technology group, so we build these AI models, then we collaborate very closely with biologists and clinicians to apply them for various questions in biology. And so I'm really going to show you first a high-level overview of how we think about this, and then in the talk I'll give you some examples and I'll tell you ahead of time so that people don't completely turn off that I've cheated a little bit and I took the whole over the human lifetime in a deconstructed way. So I will tell you some examples applying this to neurodevelopment and autism and some examples applying this to cancer. So you will get sort of through the lifetime, but not per individual. So as we think about the human phenotypes, I think it's really important that we think about those mechanisms in context. So that's a big theme in how we think about developing AI models, that this is not a generic model, but really that they can take into account deep biological context of these. And by context, I mean cell type specificity, how those cell types fit in spatially. I won't talk about this today, but we think a lot about spatial omics analysis and putting other data in the context of this, these spatially resolved methods. Then how do you put it together at the level of organs, and finally like really connecting this molecular and genomic level insights to whole organism physiology. And we also spend a lot of time about longitudinal context. So not just cell type, but what is happening at that cell type at a specific stage of development or aging. And I will show you a little bit of that. Oops, I don't know what I did. I'm a computer scientist, so I clearly am not great with technology. There we go. So specifically what that means is that we really need to be able to answer all of these contextual questions at the level of first really thinking about interpreting the genome. And what I mean by that is really thinking about the whole genome, interpreting every non-coding variant in terms of its transcriptional and post-transcriptional effect, and then obviously integrating that with the coding disruptions, putting all of that in the context of how that actually really manifests in genome regulation, be that epigenetic regulation and RNA metabolism, and then actually really bringing that all the way to being able to model networks and pathways. And finally, really integrating multi-omic analysis. And then more and more, we're really starting to think about how can we bring in dynamics into this, although I won't have a chance to talk about this today. So first, let me talk about the variant effect prediction, and that's what a lot of what my lab actually thinks about. So we build a number of models, and I'm a big believer. I won't talk about AI versus machine learning, but I will give you one philosophical point. I'm a huge believer in purpose-built models for biology. I don't think we're going to have, certainly not in the next 10 years, functional, beyond sort of basics, one giant foundation model of all biology. And I would love to talk to all of you about why I think this is really critical. But so we build a lot of these sequence models that predict specifically on the genome for any point in the genome, once you train them, a variant effect at the biochemical level. By biochemical level, what I mean is, for example, the transcriptional models will predict essentially in silico ChIP-seq, right? So our large transcriptional model will predict 22,000 different epigenetic marks, DNase hypersensitivity, as well as transcription factor binding in cell type and tissue-specific context for a number of cell types. And then we can take those effects and predict what that means in terms of pathogenicity. And what's important is the first stage, the biochemical and molecular effects, it's really purely data-driven. There is no sort of prior biological knowledge or interpretation brought in. Of course, for pathogenicity, we have to rely on what these effects would look like for known pathogenic mutations, and so there is some bias in that. But then what's important is if you have one of those predictions and you, for example, say this A2T variant in this location means that it increases expression of a particular gene early in neuronal development and it's pathogenic, now you're getting the mechanism, right, the biochemistry, and actually a context. So that's why these models are quite useful, and we have them for a number of contexts. A lot of these are published, several of them are not, but we can do that for transcriptional. So, histone marks, DNase accessibility. We have a model and review. Any of you, if you're interested, I'm happy to share the actual model. For DNA methylation, we can predict transcription factors, cell type specific and context specific expression, post-transcriptional regulation through looking at RNA binding. We recently finished a model that can go from there all the way to predicting actual isoform abundance. So there was a big theme on long read sequencing, and so this has been able to predict that variant effect on that in a tissue-specific way. And finally, really integrating the transcriptional effects into regulatory activity. So I just want to give you one example of what that means. The reason why we care about regulatory activity is that essentially, while it's really nice to get that in silico ChIP-seq effect over 22,000 different specific epigenetic factors, it's really nice to be able to tell what does it actually mean. And so essentially what that model does is it integrates those 22,000 different effects into annotating the entire genome into their regulatory classes. So that means that it finds, for example, brain enhancers, and it finds some sequences that then, once we look, actually were identified previously as brain enhancers. But it also annotates a number of genomic sequences that previously have never been annotated to that sequence class based on their similarity in these predicted effects. Now, you could have actually done that annotation by just clustering known ChIP-seq data, right, like that the community already produced. Why you need the deep learners is because now you can say, what if I have that A and it changes to a, what does it change to? A G, right? And then you can have a specific prediction that saying that this brain enhancer sequence, due to that single variant effect, is going to now lose its brain enhancer function and go into a low activity state. And then you can have a mechanism, so for example, in this case, that a particular transcription factor binding was disrupted. What can you do with them? So we've evaluated this. This has been published. The previous model has been published, so it's been evaluated extensively. I just want to show you, and this is an unpublished application, since there's so many of you coming from the cancer field, of, I think, in this community, you're probably sold on this, that it's really, we should be looking more and more at the non-coding variation in cancer. And so this is literally dividing ICGC cohort, purely taking each patient, somatic mutations, predicting their effect with that model that I just talked about. So this is purely transcriptional, and just dividing them, does it have a high impact, at least one high impact mutation next to a cosmic cancer gene, right? So we're not dividing, we're not actually taking into account known coding mutations. We are making sure this is not a tumor mutation burden-based division. Sort of all the obvious things, gene size, we've looked at, it's not just that we're putting all the, low survival cancers in one, like it's really not any of the obvious things. And you can see that there's actually quite significant and substantial difference in survival based on dividing these patients purely based on this non-coding effect. So now I want to go from the epigenetics and just show you what we can do if we take these models and train them to predict all the way to expression. And this model actually is really trying to push the boundaries of what we can predict in terms of context specificity. So part of the problem, of course, in terms of building AI model for biology is you want them to be highly specific to biological questions, but the more specific you get, so for example, in this case, we really wanted to say, what would a variant affect? How would it affect expression at a specific cell type at a specific stage in fetal development, for example? Now you're running into very few data sets to be able to train your model. And so what we developed is this new model, SEAL, this is also unpublished, is using pre-training fine-tuning framework. So essentially what it's doing is it's using all of the brain expression data to sort of learn generally how does sequence relate to epigenetics. That was the model you sort of saw before. It's a convolutional neural network model for those of you who care. And then training on all of the gene expression data, right, of the brain, which probably doesn't have, in fact, doesn't have any fetal data at all, to sort of get the general mapping. And then we actually fine-tune and we say what we really care is basically these two data sets that have profiled fetal development, where basically now we have a model that can cover about a little over 800 specific cell states, so that is specific cell type at a specific developmental stage. And so that's really interesting because now we can potentially look at cohorts where it's really important to look at neurodevelopment, like autism, and see whether we can understand the mechanisms of what are these mutations disrupted. And so we have actually done this for autism. So this is the Simon Simplex collection cohort. It's a quad design, whole genome sequencing, both parents, probands, and siblings. And in autism, there's a long-standing interest in de novo mutations, so those that you do not inherit from either of your parents. So we're looking at about 30,000 TSS proximal de novo mutations. In the past work, we've actually shown that despite the fact that there is no enrichment in the mutation count in the children with autism versus their unaffected siblings, there's actually a clear, significantly higher level of disruption using deep learners that weren't actually specific to expression, but just showing this epigenetic disruption. And that was actually, at the time, the first demonstration of this role of non-coding variants in a high level in autism. But now we can take this context-specific model and predict specifically where exactly, at which fetal stage, in which cell type are these mutations being disruptive. And essentially now we have a hypothesis that there is actually a transient fetal cell state that is specifically affected in specifically excitatory neurons in the prefrontal context, that are significantly and specifically affected in these genomes of individuals with autism. And then we can actually verify these. So I'm a little bit cheating. This is actually verification of not this specific model, but our previous model that is non-developmental stage specific, but does cover all of sort of available cell types that had previously been profiled. And so there we actually looked at variants of uncertain significance from ClinVar. And I'm just showing you P10 because even though many of you in the audience love P10 or interested in P10 from its cancer significance, we were actually interested in it from its role in autism. So hence, we're looking at our model that is specific to neuronal stem cells. That was as close before this model of gene expression for neuronal fetal development. That was as close as we could get to fetal development in neurons. And when you look at these 20 or so variants that ClinVar classifies an uncertain significance, on the y-axis is how disruptive, the prediction from our model of how disruptive these variants are, specifically in neuronal stem cells. And you can see that 4, potentially 5 variants are predicted to be disruptive, and the rest are really not likely to do anything. And in fact, when we test them, I'm just showing one result. We've tested all of these and a couple of these as controls. You indeed see a significant downregulation of P10 if you have that single letter change from the proband to the sibling variant, and it is cell type specific. So when you switch to the liver cell line, you do not see that significant disruption. And that was true across all of the variants that we tested. Now, so far, of course, as much as I love non-coding variants, I've only told you about non-coding variants and largely focused on transcriptional. I don't have time, unfortunately, to talk about post-transcriptional, but would be very happy to. But what we really want to do if we do whole genome interpretation is integrate non-coding context with coding. And also, I would really like to be able to go beyond survival and really be able to use these models to predict actual outcomes. Right, so this is where we're getting with Valeria, which is an NI framework for whole genome sequencing analysis that integrates impacts of coding and regulatory variants. The idea of Valeria is that we take, first of all, we integrate them per gene, so it's a highly structured integration. So for each gene, we're going to integrate all of the non-coding here is in blue, and coding in orange. This is also unpublished. Again, we have the framework done, so if anyone is interested in using it, we're very happy for you to use it. So we integrate for each individual all of their effects for each gene. And that means now you can have a full cohort representation of all, not just which variants they have, but how disruptive they are. And then you can do whatever other analysis that you would want to do, but now you're not having to deal with a number of the variants that you are looking at actually not being functional. So you can look at gene level comparison, you can do survival, outcome prediction, etc. So let me just go a little bit in detail for a given gene. What we're actually doing is we're running a coding effect model. This is actually alpha-missin, so it's a public model, but you can easily swap whatever you want. And we're running for the non-coding transcriptional effect model is our single cell type, sorry, not single cell, but cell type resolved gene expression disruption model. So you actually get a specific mechanism in a specific cell type where the disruption happened. And those are integrated, and you can imagine how you can extend this also for post-transcriptional regulation models, et cetera. I'll just show you one application of this. So we apply this to kidney failure. And in fact, just what's really important here is we're not trying to predict who will have kidney disease and who will not. We're taking a QRGN cohort, which is a cohort of about 2,000 individuals that are very carefully followed now for 9 years, and with 2 per year visits, clinical visits. They all have one of 4 different rare kidney diseases. So these individuals all have kidney disease. There's no healthy individuals. What we're trying to predict is for any of them, purely based on their genome, who will go into kidney failure in the context of this cohort and who will not. So this is longitudinal and really a pretty hard question, which clinically is not actually easy to predict. And we've very carefully looked at ancestry is a huge confounder in kidney disease, and this is all ancestry adjusted. And you can see that Valeria, first of all, just the fact that purely based on the genome, you can actually get a decent 67.67 AUC prediction of who will go into kidney failure, but also it significantly outperforms all of the PRS scores. And this is not just true for kidney failure and eGFR decline. You can even get some accuracy for steroid resistivity. Of course, the goal is then add the clinical information in other contexts, not just rely on pure genome, but that does show you that the genome actually has quite a bit of information, and you can really use the whole genome context to do that. Now, finally, switching from this, so far we're thinking from whole genome up, sort of thinking from the phenotype down in that context, framework of the world of the way my group thinks about it that I showed you in the beginning. Of course, the phenotypes for most complex human diseases are highly heterogeneous. So how can we actually use that heterogeneity or reduce that heterogeneity to help us discover the genetics? And I'll show you an example of what we recently finished in autism. This unfortunately lost the reference, but this was recently published in Nature Genetics, where we really wanted to address this huge challenge in autism where phenotypically it's hugely heterogeneous. And then on the genetic side, while there is very strong evidence, despite what, there's very strong evidence that it's genetic and highly heritable, and there's over 1,000 genes that have been identified as potentially linked to autism. We actually can explain an autism diagnosis for less than 20% of cases that have sequencing. And it's far less than 20% if we're very generous with what we call explain. So why is this? So what we actually did is we used this SPARK cohort by the Simons Foundation. This is about 300,000 individuals and counting. About 130,000 by now are individuals with autism. And it has a number of phenotypes, phenotypic information, and a large number of these individuals have either whole exome sequencing or increasingly whole genomes. So we ended up using about 5,400 probands because those were the probands that all had coverage over this 239 phenotypes, and the phenotypes are actually specific answers from SCQ, repetitive behavior scale, child behavior checklist, and background history. Background history is things like when did your child begin to walk, when did your child begin to talk, so developmental questions largely. We used mixture modeling to be able to account for the fact that these, of course, are all sorts of heterogeneous variables. And we found that no matter whether we used computational techniques or actually our collaborators, clinical psychologists, Jennifer Foss-Feig and Leanne Snyder, also looked at it from the perspective of sort of clinical resolvability for subclasses, really very robustly was what came. And we actually did replicate this completely in an independent cohort, the Simon Simplex collection I mentioned earlier. This cohort SPARK is individual or parent reported. Simon Simplex is fully clinical. They're actually quite different cohorts, and the model replicates incredibly well. Whether you just take the model trained on SPARK, apply it there, or if you use the same process and train a new model in SSC and then correlate the classes. So here are the 4 classes that we found. I'll start with the green subclass. I'll start with the x-axis. On the x-axis are basically clustering of these 239 phenotypes that we used. We did not use this clustering for any analysis. This is purely to show you instead of showing you 239 points. And this is our clinical psychology collaborators just grouped them into these sort of 7 axes of, anxiety, mood, all the way to developmental delay. So this is how these features mapped. And then on the y-axis, we're showing how much individuals in a particular subclass were had the challenges in that specific axis. And this is with respect to other individuals with autism, not with respect to unaffected siblings. I have a graph that I don't have in this presentation showing that even this moderate challenges subclass that you can see appears like it has no challenges. It has moderate challenges that are significantly more affected than unaffected siblings. So let's look at the green subclass. That's the social behavioral subclass, and this class is characterized by pretty much no developmental delays. These children develop just like their unaffected siblings, but they actually do have significant social and behavioral issues, and then they have significant challenges in the context of anxiety and repetitive behavior. And this is in contrast to the blue subclass, mixed autism with developmental delays. These children have significant developmental delays across all spectrums, significant levels of intellectual disability, and almost no anxiety and attention challenges. And then I already told you about the moderate challenges subclass, and that's in contrast to the broadly impacted subclass. These individuals have significant impact basically across every single aspect of this spectrum. Now, once you have those subclasses, does this tell us something about the heterogeneity of genetics? Because that's where we came from. And it turns out we were actually extremely surprised, to which, I mean, we started this, we kind of expected some differences, but to which extent these are very different on biology. There's almost no overlap in biology of these four subclasses, very limited. I won't show you the common variants. I'm happy to show it to you later if you're interested. But for the rare variants, so one is the similarity. So not surprisingly, the community knew this. De novo rare variants of high impact are enriched in every subclass, so that is true. But here is a big difference. When you look at the mixed autism with developmental delay subclass, that is the only subclass that is significantly enriched for high impact rare inherited variants. And this is really important because previous studies, so of course we're not the first ones to realize that autism is heterogeneous by far. Anyone who has anything to do with autism knows this. But previous studies really focused on specific axes, nearly always either an axis of intellectual disability or the axis of motor skills, or both. And essentially, if you look at that axis, so for example, intellectual disabilities, the levels of ID in those two subclasses, in the pink, broadly impacted, and the blue, mixed ASD with developmental delay subclass, are pretty much identical. They're completely indistinguishable. Yet you can see here, not just the biology, the pathways that are impacted, by the way, in these four subclasses is totally different, including those two. But here you can see even the mechanism of mutations that are playing a role is different. And then I wanted to focus on another aspect in which they were really different, that's development. So what we can do, of course, we don't have developmental gene expression samples from individuals with autism. What we do have is we can take the genes that are affected by these variants and map them to a fetal gene expression data set in presumably normal individuals, right? And so what I'm looking at here is the classification that came out of the study. So Herring et al., sorry, this is our paper that's been published for a while. I just didn't switch this. Herring et al. Is a data set of fetal gene expression, right? And that data set basically divided, for our purposes, the genes, and they divided into four subgroups, but really for our purposes, there's genes that are primarily expressed postnatally in yellow, and genes primarily expressed prenatally in blue. And so now we can essentially map our variants for each subclass and say, where is the significant disruption happening? And of course, we're making an assumption that if a gene is not expressed, probably that disruption, obviously the mutation is there, but the disruption is not really active yet. And of course, if you look at siblings, nicely, we can see that there's no enrichment. If you look at all probands, not surprisingly, we see a very significant disruption. So sorry, you can see the fold enrichment here, and then whether it's filled in, the circle is filled in, just shows if it's significant. You can see the very significant disruption across a number of cell types for prenatal genes, and this one showing up here for postnatal. Here's where really the surprise comes in. When you look at the social behavioral subclass, there is no significant disruption prenatally. There is nothing happening, at least nothing significant. So disrupted genes are really most active post-fetal development in this subclass. And then if it's extremely contrasting to the mixed ASD with developmental delay subclass, where now you see pretty much all of their disruption is happening prenatally. So that means very different potential trajectories. And in fact, if we look clinically at the diagnosis age, indeed, that is completely reflected in the diagnosis age, where the kids in the mixed autism with developmental delay subclass are diagnosed very early and significantly earlier than the kids with the social behavioral subclass. Of course, that has all sorts of implications for understanding how to support these children, what to tell these families potentially in terms of their trajectory, et cetera. So that's it. Really just wanted to highlight that it's really important to look in a high context with these AI models, and to be able to look across basically the longitudinal context, the treatment context, and the dynamics. And I do want to make a shameless plug for humanbase.io, where we share most of our tools. You can actually upload variants and get a lot of our models are implemented there in a very easy-to-use framework, and you can download any of these networks that I didn't have time to talk about, but that are quite useful to analysis. And that's the people who actually did the work. I just go around and give talks. So thank you.